Now let's talk about ultraviolet and visible spectroscopy. Usually we deal with these two in the same section because they're kind of in the same region, and also you require only one lamp in order to do this spectroscopy. Okay, so in UV-Vis spectroscopy, we deal with a region of 200 to 400, which is the UV region, and 400 to about 800, which is the visible region. And they're done together, as I said previously, they're done together because one lamp can provide enough energy from 200 to 800 nanometers. So in any case, what happens in this region then between 200 to 800 is that the energy that is given to the molecule, those photons will then excite the electrons from a bonding orbital to an anti-bonding orbital. And we'll talk about this a little bit later also. And we've talked about this before in conjugation, if you can remember this, okay? And so in any case, the electron transitions are going to be occurring in these cases, all right? So in IR spectroscopy, there's vibration going on. But in case of ultraviolet and visible spectroscopy, there is electron transition going on from a bonding to an anti-bonding orbital, and this is because UV-Vis region is higher in energy than IR, okay, and that's why these kind of transitions can occur. So as we talked about in conjugation, also conjugated dienes or conjugated compounds have molecular orbitals that are closer to each other in energy, and so which means if they're closer in energy to closer to each other in energy, then the electron transitions can occur a lot easily. If they occur a lot easily, they don't require so much energy. If they don't require so much energy, then you will see the absorption in more of the visible region than in the ultraviolet region, because remember, the visible region is lower in energy than ultraviolet. So here are some examples. This is a 4-carbon system, this is a 6-carbon system, and this is a 12-carbon system, okay? And so if you look at all of these compounds, which are all conjugated systems, you can see that as the conjugation is increasing, so is the lambda max. And the lambda max is the highest absorption that you would see for a compound. And I'll show you a spectrum a little bit later. And so the lambda max moves from 217 nanometers to, in this highly conjugated compound, to 364 nanometers. It's almost a jump of about 150, okay, nanometers. So conjugation can have that effect on where a molecule will absorb in the visible region or in the ultraviolet region. So lycopene, for example, which is found in tomatoes, okay, it says, why is it red? So look at the conjugation that's going on here, okay, except for a little break here and a little break over here. Lycopene is super conjugated system, okay, so lycopene absorbs in this part of the visible region, so which means this is not absorbed. And so if something is not absorbed, that's the one that you see, okay? And you might have seen color wheels, in which case the opposite regions are the ones that you see, okay? So if one color is absorbed, the opposite one in that color wheel is the one that is actually seen. So in any case, this is what happens to lycopene, okay, and how it absorbs at lambda max 470. If you remember, the other one was 364. So lycopene with more conjugation now is moving now into the visible region, okay, 470 is the visible region. So this is a refresher for conjugated systems and all that we talked about as far as the molecular orbitals are concerned. Okay, so in molecular orbitals, if you remember, you had the positive regions and the negative regions, which are the negative and positive wave functions. And so if the positive wave functions are all on the same type, on the same side, then they form what is called the bonding molecular orbital. And as you go higher in energy, then the molecular orbitals can have nodes in them. Okay, and once they have the nodes, it means that they have opposite regions next to each other. And so by the time you get to the top one, which is the highest energy level, there are maximum number of nodes, which means that almost all the orbitals are in opposite to each other, okay, or is an opposite charge to each other. And again, the positive and negative has nothing to do with the charges. Remember that it is a wave function, okay, and so it's a matter of just forming nodes, okay, and nodes are a region where you don't find electrons. Remember that too. So in any case, when a compound is at a ground state, you will find all the electrons in the bonding region. Okay, so all the electrons will be distributed here as much as possible, okay, in the lower region. So what happens then is when you provide energy, say in the UV region or in the visible region, or you provide energy to a molecule, if it is possible, okay, then electrons are going to jump from the bonding region to the anti-bonding region. This is the anti-bonding with a little star on it. And so they'll jump from the bonding region to the anti-bonding. Now they don't go from the top to the very very bottom to the very top. They actually go from the highest occupied molecular orbital, HOMO, to the LUMO, which is the lowest unoccupied molecular orbital. So which means the transitions will start from the closest orbitals, and then the next one will come over depending on the energy and depending on the orbitals. These orbitals that we find are going to be for every orbital in a molecule. Sigma bonds, pi bonds, non-bonding electrons, all sorts of orbitals possible will be shown over here. That is the beauty of molecular orbital theory, is that you will account for every single orbital that is present in a molecule. So which means that the more HOMOs and LUMOs you have, or the more of these bonding and anti-bonding regions you have, the closer the HOMO and the LUMO become. And so the closer they become, the easier it is for the transition to occur for an electron from the HOMO to the LUMO region. Okay, and so that is why you can see molecules absorbing in the visible region if these two are close to each other, and in the UV region if they are farther away from each other. So when conjugation occurs, when there are lots of non-bonding electrons, then you have many, many orbitals, so which means they start getting closer together. Okay, so that is kind of the fundamental behind UV visible spectroscopy. When you study a little bit more on what this is called like photochemistry. So when you study more on photochemistry, then you learn a lot more about what kind of transitions occur, and not only what transitions occur, but how do these electrons then come down, okay? Because once, of course, something goes up, it has to come down. So if something electron is going to a higher energy level, it eventually has to come down too, okay? And so you study a lot more in detail about how the electron transitions are occurring in photochemistry. Here is an example of what happens when you have conjugation and the molecular orbitals. Okay, and so we looked at a little bit about these systems before also. So you have a 4-carbon system, 6-carbon system. This is not 12, but a little bit more conjugated than this one with 8 molecular orbitals. And this is kind of what happens when you have more conjugation going on. As you have more conjugation, you have more pi bonds, and so all of these molecular orbitals start moving closer to each other, okay, which means that the energy between them gets progressively smaller and smaller, and that's why the electron transitions become easier to do. And so these kind of compounds with more conjugation will then start absorbing in the visible region rather than the UV region. All right, and so this is kind of what goes on. Of course, these are simple molecules. When you have larger molecules and lone pair of electrons, these diagrams are complicated, and the electron transitions also are very complicated then. So how do you obtain a UV-Vis spectrum? The instrument is almost the same as an IR instrument, okay, which means that you need to have a source of energy, and the lamp here, remember, is going from the ultraviolet region all the way to the visible region, so it's going from about 200 to about 800 or 900 nanometers. You need to have two reference cells, or two cells over here, which means that usually this instrument is dual beam, as we call it. One has to be the reference cell, the other one has to be the sample cell. The way we make the sample for ultraviolet is that you actually have to dissolve it in a solvent. So when you dissolve it in a solvent, you have to nullify the solvent, any absorption in the solvent, and so therefore you need to have the reference cell with just the solvent. If you're looking to buy these solvents and you start looking into the catalogs for buying the solvent, you will see that some of the solvents are already made for UV vis, which means that they don't have any particles in them that will absorb any light. Okay, so it will say that this is spectra grade or whatever grade it is, but it is still a good idea to go ahead and keep a reference cell. to eliminate any kind of absorption from the solvent. Okay, and the typical solvents that are used are dichloromethane or methanol. Those two will pretty much dissolve anything that you have in your lab. Okay, so use these solvents and you make a dilute sample. You don't make a concentrated sample. Like for IR, you use neat sample. You don't have to dissolve it in anything. But in case of UV spectrum, you really have to dissolve it in something. You have to have a container. So the container is usually called the cuvette. And in some cases you can use a test tube also, provided the glass does not absorb in the visible region. So usually a quartz test tube is used, or a quartz cuvette is used, so it does not absorb in the visible region or in the UV region. All right, so glass will not absorb I think in the UV region. Okay, but the quartz, it's really very good. Quartz does not absorb anywhere, so it's much better to use a quartz as your container, and then you take these solvents and then nullify them by using the reference cell. Once the beam passes through, it goes to the detector, and then of course you have your output, okay, which this is what the spectrum might look like, which we'll see in the next slide over. Okay, and so this is your very simple kind of an instrument. Okay, and don't worry about this so much, really. This just tells you that how we find the absorption, okay, which is the intensity of the solvent, and then of course the sample. All right, which means you're trying to nullify the solvent absorption in this case. You need a really very small amount of your substance. You don't need a large amount of substance because the scale is very limited. So this is really what a spectrum looks like. You don't really have any kind of clear peaks in this. You have broad peaks. It's not like IR. Okay, in case of IR, you're able to see the functional groups and you know exactly where to look at. You know what you're expecting. But in case of UV visible spectrum, you really don't know what you're going to be seeing. Okay, when you make a new compound, you can sometimes not really predict. In fact, you can almost never predict what the spectrum will look like. So only when you run the spectrum, then you get to see what it looks like. Okay, the scale for the UV spectrum is absorbance versus the nanometers over here, which is lambda. Lambda is the wavelength. Okay, and so you can go from 200 all the way to 800. If you're looking at the visible spectrum, you will start at 400. If you're looking at the UV spectrum, you will start from 200 and go up to 400. So isoprene over here, this one absorbs only in the UV region because it doesn't have that much conjugation, only 2 bonds here. So it absorbs in the UV region, and this is what you end up getting. So the absorption scale, this is the one that you really have to mind for, because in case of IR, you didn't really have any issues. It was just transmittance. But in case of UV visible spectroscopy, you have to be careful because the absorbance scale is very limited. Okay, you can go up to 10 also. But the thing is, if your sample is too dilute, then you may not be able to clearly see the lambda max. And on the other hand, if it is too concentrated, then this peak will be so high up there that you don't really know where your lambda max is. Okay, and if it's too dilute, it will be way down here, and then you don't know what your lambda max is. So you really need to have a nice concentration of sample in there, and sometimes this takes a little trial and run, and then you figure out, okay, this is the concentration I need. So if you're looking at colored samples, then you try to make sure that you have some color in your sample, but that can be very deceiving also. All right, so this is something that you really just have to work out as you're taking the sample, as you're taking the spectrum. Anyhow, so you get the lambda max only from the graph. There is no other way to do this. Okay, and so you read the lambda max just by reading the wavelength at which you have the highest point. In case of isoprene over here, you only have one lambda max, but depending on what kind of a compound you have, you may have more than one lambda max. It is not that you'll just have one peak. Sometimes you can have more than 2 or 3 peaks also. Okay, it's not going to be as complicated as IR though. It's still quite simple, but if you have more than one peak, then you have to mention all of those lambda maxes. One of them usually will be the largest one in any case. The absorbance usually follows what we call the Beer's law. Beer's law states that the more concentration you have, the more absorption you have. Exactly what I was just talking about is that is why you need to make sure that your sample is not as concentrated. Okay, so absorption is directly proportional to concentration. This is Beer's law. This is absorption. The epsilon over here is called molar absorptivity, or it's also called extinction coefficient. and C is the concentration of the sample, and L is the length of the light, okay, because it does matter how much of light is passing through your sample. So in case of most of the cuvettes that we use, they're all 1 centimeter. So we don't have to worry so much about the length of the sample. We have to just worry about the concentrations, okay, and so on. The epsilon over here, the extinction coefficient, has to do with the area under the graph, okay, which is this over here. So it tells you how many electrons are capable of transition, okay, at that particular wavelength. So for example, in this case, there are the maximum number of electrons doing the transition at lambda max of 220 and 22. If there was a small bump over here, then you can say, oh, there are a few electrons that can do a transition at the 300 nanometer wavelength. So the extinction coefficient gives you a measure of how many electrons are actually doing the transition. All right, so that's what this area under the graph is, or the extinction coefficient is. But anyhow, this is what a UV vis looks like, the spectrum looks like, and it's really not very glamorous. Here are some sample absorptions. I don't need you to remember any of this stuff. I'm just giving you so that you have an idea of what things are like and how they absorb. And this is just with one bonds or isolated double bonds. These are a little bit of more conjugated dienes, and if you look at the conjugated dienes, you can see how they absorb. So there's not so much difference between the butadiene and the hexadiene, where you have the 2 double bonds, and then here is the cyclohexadiene. Now remember, with all of these double bonds that are occurring here, why is there a slight difference? It could be due to what kind of an overlap you have. Here you have a ring structure, here you have everything in a straight chain, and then here this is going down. Once you have the double bond outside and inside, then an external and internal double bonds, this is going to be slightly different because of the way the overlap is. So the conjugation is still there, but not as good as it was over here, and so therefore the absorption can go down. There is a way to figure out that if you add groups onto your compound, what kind of an absorption you will get, which are called Hoffman rules. So once you get into a deeper UV vis spectroscopy, then you can figure out, okay, what happens if I add an alcohol group? Will my absorption increase or decrease? And it should increase in the wavelength because you're adding lone pair of electrons. Okay, so what happens if I add another conjugated system? What if I have another double bond over here? Will my absorption increase? And it should increase because you're increasing conjugation. By how much? So maybe about 20 nanometers, whatever it is, but those are called Hoffman rules. And you get into those when you, again, as I said, when you get deeper into UV vis spectroscopy. Here are some more conjugated dienes and trienes and compounds. And then the dyes. In amines, we talked a little bit about dyes and the fact that you have highly conjugated systems, okay, in dyes, and that these dyes, they absorb in the visible region, which is why you can see them. If things don't absorb in the visible region, you will not see them, which means that they will be colorless. But if something absorbs in the visible region, that's the reason it has actually a color, okay, in there. And so methyl orange, for example, okay, it absorbs as 440 nanometers. And again, these absorptions are due to the highly conjugated system. So if you have a highly conjugated system, then those HOMOs and the LUMOs are very close together, and so it doesn't take that much energy to do the electron transition. Okay, here is indigo, and remember we talked about indigo before, and even if we have not, it doesn't matter because here is the conjugated system, highly conjugated, so many lone pair of electrons also present over here. So all of this causes for a really good transition between the HOMO and the LUMO. Look at this one, Congo red. Okay, it's highly conjugated system. So that's what happens with dyes. Most of the dyes that you will look at the structure of, you will see that all of them are conjugated systems. I added this one here for personal interest also, and just also because it's so much fun to see. I used to work with porphyrins before, and so I always find them very fascinating. They're huge molecules to work with, and they're very colorful. Chlorophyll is usually very blue-green in color, and then of course the color of the leaves happens in the chlorophyll degrades, you know, and so on. And then of course you have the hemoglobin, where you have the heme, which is also a porphyrin structure, but that's red in color. So you have such beautiful colors with porphyrins. So I thought I'll just show you a spectrum, okay, of what a porphyrin would look like. And this is chlorophyll, and chlorophyll, this is chlorophyll A and B. They're slightly different in the structures. You can see that over here with the aldehyde here, no aldehyde here, and so on. But anyhow, chlorophyll A, chlorophyll B, they have these bands over here around 450, so they are highly absorbent in the visible region. And then look at that, they also have bands over here. They also have small bands here. Okay, so the blue line that's going here is chlorophyll A, and that's because chlorophyll is slightly bluish in color. That's why they have the spectral line as blue. And so this is the absorption, and then you can see that there is so much electron transition going on even around 670 nanometers approximately. And then chlorophyll B, which is more of a green colored compound, this is where the absorption is, okay? And so then you can calculate the extinction coefficients of each of these bands and see which one is more efficient in absorbing light. The yellow one is carotenes. Carotenes are like what we talked about before with lycopene and all that. That's kind of what carotenes also look like. They're reddish or yellowish in color. And usually what happens in fall is when the leaves start changing color, is that the leaves actually stop doing photosynthesis, and so the chlorophyll will start degrading. And once the chlorophyll is not there, the color goes away and the carotenes start popping up, and then that's why you see the yellows and the greens, the yellows and the reds, is because of the carotenes, and some of it is also because the chlorophyll, when it degrades, it still gives some different colors in there. So anyhow, this is what the absorption spectrum of chlorophylls look like. Very, very colorful compounds. So the key concepts that I need you to know from this one, from UV-Vis, is that you first should definitely be able to recognize conjugation, which should not be hard to do because we've done this before. So once you recognize conjugation, then you should be able to predict which molecule will absorb more in the red region or the violet region. The red region is the visible region, the violet region is the UV region. Okay, so if you have more conjugation, it will absorb more in the red region, in the visible region. If you have less conjugation, it will be in the UV region. Okay, so those are the main things. Other than that, you really can't predict much, okay, in UV-Vis spectroscopy.